Whole genome sequencing is the beginning of a new era in the diagnosis and treatment of cancer. And here at the 100,000 Genomes project, we've been working with the NHS Genomic Medicine Centres to spread its use across the NHS.
So the purpose of whole genome sequencing is just that, to obtain a whole genome sequence from a patient's tumour sample and also from their germline DNA. Rather than just looking at a tumour as we do now down the microscope, you can also now assess the genomic changes, and you can identify changes that may be targeted by certain drugs.
Cancer is a genomic disease, so if we can study the different types of mutation in the whole genome of the tumour, we can get very useful information that tells us about how that tumour is going to behave. So there is prognostic information. In addition, there are a number of research trials underway which are specific to patients with different types of tumours. So studying the whole genome will enable us to see if patients are eligible for different research studies and will help us recruit them.
We're beginning to get the patient sample details back, so the sequences are beginning to come back from patients who have been involved in the 100,000 Genome project, which have been useful in a couple of cases in terms of allowing us to target their treatment for their cancer type. But in addition to that, the results from the project are going to be useful in a more wider field in terms of the research that will be carried out to allow researchers and clinicians to start developing new treatments for the future, which will be for patient benefit. And the other thing that's come out of the 100,000 Genome project that I feel locally is it's added to your multi-professional working. We've built up and solidified working relationships with people across teams within the hospital. And another benefit that has been very important is bringing together those teams in order to push forward education in molecular pathology, which will be useful for developing our future pathologists.
For the best sequencing results, DNA needs to be fresh, and this has presented some problems. One of the most important has been overcome recently with a consensus statement from the Human Tissue Authority, the Health Research Authority, the Royal College of Pathologists, Genomics England and NHS England. It enables samples taken and kept fresh for sequencing to be considered diagnostic up until the point that the results are returned to the clinician.
The HTA consensus statement is an agreement that we can collect appropriate tissue, and for that we typically would mean fresh frozen tissue, in order to undertake genomic analysis as standard of diagnostic care within the NHS. And that's very important because historically collection of fresh frozen tissue has very much been for research. So you've had to consent the patient up front in order to process their tissue down a fresh frozen pathway. And therefore, if you haven't had that consent up front, you can't process the tissue down that pathway. The HTA consensus means that actually, because we appreciate that we are at a point where molecular pathology and genomic studies are part of the way that we should be treating patients routinely, that accordingly we should process the tissue optimally, and that's in a fresh frozen pathway, to enable those types of analysis as routine. So that can all happen as part of their standard consent. And then if we want to use their samples or data for research, we can then consent them downstream. And that's been very useful for helping recruitment to the 100,000 Genomes project.
The other problem is formalin fixation. Formalin damages DNA and may make the sequencing results inaccurate. Routine processing methods need to be adapted, and in this series of videos, we'll be showing you techniques both for large and small tissue samples.